last class, already we have the cell biology. The cell biology, we know the importance of cell biology because later we are going to study about the genetic engineering and somatic hybridization quite very important. In the cell biology, I have told that's a big difference in the plant cell. Already we have studied the plant cells and animal cells. We have studied including the virus, bacteria. We have learned from the microorganism. Then the plant cell, we are very much concerned about the plant cell in case of better biotechnology. So what is the important characteristic plant cell is plant cell wall. Last class we have seen, and we understand that there are some different types of cells and the importance of the cells, and they are rigid. The primary cell, the middle lamella and pectin cellulose, primary cell wall, the upper one that is like a cement, middle lamella. You want to study the middle lamella later, then only you can get it. Then the plasma membrane, then there's a component, cell wall component. But this one I don't give you notes. You have to write again and again. I better to write by your hand. Handwriting is very important. Otherwise, it may be very difficult during the, and especially if you are the first year students, each and every spelling. That's what we are teaching this one again. You already studied this one even in the O level, but we are studying because to slowly we are improving English knowledge. So please, whatever it may be, you write. Take a note, small. And last class we have studied these are the carbohydrates, they are monosaccharides, they are the building blocks. It's very important. You can get it anywhere notes. Even I have cell wall. You have the structure and they are the pectin, cellulose, molecular fiber. This class we have studied, and especially in the plant cell. Why the plants are trees? So very strong compared to animals because of their plant cell wall. Especially within the plant cell, the lignin, which is found in the secondary wall, these are the very important things that last class we have seen. So these are the important things. If you have any important features, you have to write. And I have told last class what is the special properties of plant cell. You can go in that book also there. One of the cell wall, chloroplast, the big vacuole. So if you miss a cell, I have gave the books. If you miss something, you can go and learn more. Okay. Then I about the cellulose also. You can put it just in the not cellulose. If you couldn't write it each and everything, later you go and refer. That's the attitude you have to develop, at least in the university. Already in the school, they already dictate the notes by one by one. You will write and you will memorize and you will expose in the examination. It doesn't develop your personality. So that's very important. So please, you miss it something, you can go and find this up. Then ours everybody has even the Google or anything, you can get it. Just few things, understand and write it and make it the clear one. Then we have studied the cross linkages, space, plasma desmota. This is pectic polysaccharides. I think you will learn the whole chemistry about the polysaccharides, pectic polysaccharides, monosaccharides, and all these things. We can repeat it again and again. Then the fourth thing. So if you visit just the pectic polysaccharides, put a small title, leave 4 or 5 lines, then later you can fill it. Don't worry, so it's going fast. So these are small things we have to learn a lot. You know that cell biology, I said the book 535 pages. If you want to study all the things, how many Yes, you need. If I want to teach that one, even that cell biology, 535 page, it needs 4 years. So you will tell the important points and you can move. The proteins, the both proteins are typically glycoproteins. I think you have studied already. We will come discuss. The both proteins also have the structural roles. Since the amino acids are characteristic of other structural proteins such as collagen, later we will start. To extract the protein from the wall requires destructive conditions. We will study today if you have time. DNA, nucleotide, DNA, then how amino acids, proteins, enzymes, genome. If we have time, we will go through today. Then say lignin, suberin, wax, cutin, water. These are the things already we have studied anyway. You have to understand that plant cell wall is a very important one, which is gives strength to the plant or trees, and especially within the structure, function, composition, special characters like the lignin. Lignin is a special character here. And this lignin not only gives the strength, but it resists the fungal and pathogen attack. So these are the things you can note it lignin, suberin, waxing, water. If you visit something, you can later you can add it. They are thicker than the plasma membrane. You will study the later animal cells. Yes, plasma membrane, but this is thicker, and you can see that's a primary. Well, there's a pectin and cellulose microbial. Last class we have drawn. Then middle lamella, primary lamella, secondary, tertiary, the middle lamella. We have learned. Then plasma desmata, it function as the interconnection between the cell and this one, isn't it? The middle lamella is formed during the cell division, and accordingly, during this one also you will learn in the cell division. Then the cell wall have primary cell wall, which accommodate the cell as it grows, and the secondary cell wall. They develop inside the primary wall after the cell has stopped growing. During the primary cell wall, they will develop when you grow, but after the cell growing, the cell division stop, then secondary cell wall they will develop. That's the primary cell wall, secondary cell wall. In the secondary cell wall, the lignin will be there. So while growing, it's very not strong. Then thereafter, after growing that one, then the secondary cell wall will form. That will give the strength to the plant cell wall. And then you can go to the one is the primary cell wall. What other things are incorporated there? Made up of cellulose, chemicellulose, pectic substance, lipids, proteins, mineral elements, and water. Everything is there in the primary cell wall. It is next to the middle lamella. Middle lamella means that's a two like a wall. They will put it down in the middle one. They will put it cement, that middle lamella. Okay. Then there's a composition. Composition of the primary cell wall as usual. Pectic polysaccharides, chemicellulose, cellulose, protein. No need to. The percentage is not necessary for the exam. But if you know the pectic polysaccharides, chemicellulose, cellulose, proteins are in the primary cell wall, that is enough. That's just for the composition. Then the primary cell wall is thinner and more plain than the secondary wall. Secondary cell wall is important one. That's exactly they will give the this one. The main chemical components of primary plant cell wall include the cellulose complex, carbohydrate, and this one. In addition, the cell wall contains two groups of branch polysaccharides, peptides, and cross-linking glycans. Maybe it's new to you, but it's okay. Whatever you can mention it. Later we will study later also. Cross-linking glycans increase the tensile strength of the cellulose, whereas coextensive network of peptides provides the cell wall with the ability to resist compression. It's okay if you sometimes okay. Just I am telling this one, you can able to get it. get it. Otherwise, we can dictate. You go through that book also. You can get it, and especially for your term paper presentation. I think Neroshamis have told about it. They will give the title earlier. Students are less than 50, then we will give each title for every student. Then they have to prepare a PowerPoint slide and they have to submit the assignment as a term paper. This one about around 5 to 10 pages. Sometimes this, some student 30 pages they will write, and you have to present that one. But this times, I think for 3 students, 1 title, and I tried to get the title because you don't have the books now. Okay. Then the secondary cell wall. This is the after when growing the cells. So during the cell division, primary cell wall they will develop after the of the cell division. So then secondary cell wall inside the secondary cell wall it has the lignin, which is deposited in the lignin will give the strength. So that's the secondary wall. So the term paper title I because usually you have to go to the library and get the 4 or 5 books from the 5 books. You have to collect information about the title, but you don't have books and you don't have the opportunity to move the library. Therefore, one book ebook you have. We choose the title from that ebook, and you can go and get and try to do the presentation. Okay, so this is the second cell wall. Cell wall is deposited in lignin. So that's an important thing. That's what that lignin deposited lignin, an additional layer inside the primary wall. This occurs after growth stops or when the cells begins to differentiate. The cells earlier they will cell division will take place, cell multiplication or cell division. Then during that time we call this cells are undifferentiated cells. Even later we will study in the third year first semester, we will produce the callus. Callus are undifferentiated cell mass. Undifferentiated means their function is not yet determined. You can use it those cells for anything, for shoot induction or root induction or some other purpose you can. But differentiated means its function is already determined. Whether we root or shoot or thereafter we can do it. But here, before that, before this occur after growth stops or when the cell begins to differentiate, when it started to differentiate, the function is determined. The secondary wall is mainly for support and comprised primarily of cellulose and lignin. Often they distinguish. So this is the important thing. One. And about that lignin also you can write because the center of the plant comes from the plant cell wall. So within the wall, the secondary cell wall. Within the secondary wall, which is important, the lignin. So we have to mention something about the lignin. Lignin is the general name for the group of polymers of aromatic alcohols that are hard and impart considerable strength to the structure of the secondary wall. So if you can write it or take a screenshot or anything, or otherwise you can get it from the. But you should know plant cell wall, structure, composition, important features, function. The important features mean here the second cell wall and the lignin. This is what provides the favorable characters of wood to the fiber cells of woody tissues. It's also common in the secondary walls, xylem cells, which are central providing structural support to the plants. I don't go over the deep because even the first year first semester it's difficult. But really, yeah. But nowadays you can escape. Earlier, usually students come because of the English. They will come within 5 days. They will come Monday morning and Friday evening. Until that one, they will wait, and they will run to home. They don't return back. So we don't give much stress. It's okay. Lectin also make the plant cell walls less vulnerable to attack by fungi or bacteria. Will give the resist as do cutin and suberin. Another, okay, this one you can mention that one. Then tertiary cell wall. Tertiary cell wall is deposited in the pure cells. It is considered to be dry residue protoplast. That's together. Besides cellulose and hemicellulose, cylon is also present. So the primary cell wall, secondary cell wall, the lignin about the lignin and tertiary cell wall, and how it's function. The structure already we have. That's all. Same as like this one. Each and everything. If you miss something from the during the lectures, you can get it here after cell wall. We are going to study one by one. The nucleus, then mitochondria, chloroplast, each and everything you should know. You can develop your notes. You can get it from the mitochondria, chloroplast, lysosomes, each and every endoplasmic reticulum. Structure, their composition or importance, and their functions. Structure and functions are important. If you get it, all these things, that's also finished. Cell biology is okay. All this, the nucleus, chromosomes, inside the lysosome, endoplasmic reticulum, mitochondria, chloroplast, isn't it? What is about the cytoplasm, everything, and thereafter cell division, mitosis and meiosis. That's what we are going to study here. In the nucleus, I will take about the nucleus later. We will study, but within the nucleus, the DNA. How DNA, nucleotide from the DNA. Then, so these are those things are. I will explain that you will understand what is a DNA and RNA. Today, if possible, we will start that DNA and RNA, or otherwise we can go for another day also. Doesn't matter. Then the functions of cell wall. That's everything. In all the organelles, you have to find. You can get it from the notes. You can make it very easy, and no need to hard. The points form. The first year of semester you can write in the points form is okay. So maintaining or determining the cell shape. That's important. That's cell wall that determine the shape of the cell. That's the shape and protection. That both are, and the communication. These are the basic things you have to understand. Maintaining or determine the cell shape and external skeleton for the every cell. Since protoplasma invariably round, this is good evidence that the wall ultimately determine the shape of the plant cell. If you break down the cell wall, you get the plant cell, and if you break down the cell wall, that is protoplasm. After the breakdown, it will become a round shape. So, when the cell wall, it gives the shape. Then support and mechanical strength allows plant to get hold out thin leaves to obtain light. So it will give the support, the maintaining down, determining the cell shape, then support and give the mechanical strength. That's what I have told. Then prevent the cell membrane from bursting in a hypotonic medium. When you put it in the high pressure or concentrated medium, it will protect. Then controls the rate and direction of cell growth and regulates cell volume. The volume of the cell, it will be determined by the cell wall. If the cell wall is to become this, then we can get the more volume. So these are the function is important: structure, function, composition, special features. So each and every organs you have to study, including the plant cell wall. Okay. Then ultimately responsible for the plant architectural design. How it's going to which direction, how it's going to look. The plant, everything is decided and controlling the plant morphogenesis. Since the whole dictate that plants develop by cell addition. So this if you want to learn more about the functions of the cell. You can go and get in that photo. Then the carbohydrate storage, the components of the whole can be reused in other metabolic process, especially in the seeds. We will take the seeds, we will come later also. So this cell structure and function is you can, and the physical barrier, even the pathogen or anything, when it covers with the cell wall, it's a physical barrier. They don't allow to enter some of the things into the plant cell. So it will the protects. We have learned it resists the fungi and bacteria, especially the lectin. However, remember that the wall is very porous and allows the free passage of small molecule including the protein, even though as the passage is there. Suppose about 4 nanometers. So, okay, take a recognition for the example. This is even within that they have the, it's okay. When the economic products, cell walls are important for the products such as paper, wood, fiber, energy, shelter, and even roughage in our diet. So, because of that cell wall, we are using for all these things and has a metabolic role. Some of the proteins in the wall are enzymes for transport and segregation. Even the plant cell wall, that's enzyme. We don't need to go for such deep level at first year of semester. We will come again. Don't worry. Then this is other activities, but not necessarily. I don't going to fetch too much of things, but you can use oligosaccharides, which can result from the normal development of pathogen attack, serve a variety of function, including stimulate the ethylene synthesis, induce phytolexins, but it's okay. Even if you don't know this one, it's okay. But you can, by your own, you can get your notes that would be better. Try by hand writing will definitely. At the meantime, because of I don't know, we have identified different way to have your conduct your final exam. If you are ready, we'll have next week. We'll have the quiz exam by online quiz exam through the computer. We'll tell you. You can tell your opinion by that. Anyone can you say? Did you have any exams earlier with the heat? Did you have any exams? Quiz exam or some other? It's okay. If you like not only the cell biology, if you like that one, okay, they have put it in the chat box. It's okay. Okay, thank you. Hello, Shamil. This will tell you how to do the exams through the LMS. If you agree with us, we can. Samina, it's okay, no? We will have next week. Okay. Prithvi. Okay. Very simple. Okay. You can go through learn this one, and even from the some lectures from the. Which chapter? Until don't worry about that. Until this plant cell wall. Cell wall or anything. This is a fourth week or fifth week. Now you are covering. You'll find. You can everything you want to put it in the chat box. Okay, sir. Which chapters will come? Okay. I will tell you today's Friday. Anyway, just you learn. If possible, we will have the exam on next Friday. That is 1st of October. Until this cell wall. Even from Botany also learn a little bit earlier things. I will tell you. We have okay. We will move here. Yeah, it is here. And these are the things. It's too much. It's okay. Let's look at how the system works. Consider the pathogen fungus like phytophthora. It's okay. We can be ball formation. How the ball is formed. Charali, cute. Okay. These are the things. Enough. If we want to learn more function about that one, we can learn later also. Okay, that cell wall. I will stop here. The cell wall. Other cell organelles, they will teach you later. I will come this one. Oh, 133. So, next Friday, 10:30, be ready for the exam. But if you prefer, if you don't like it, it's okay. But get into touch with something. That's good. So, the 1st of October, Friday, 10:30. But 10:30, can you do your exam early morning? Everyone is putting the chat box. Which chapter will come? So we have to study practical notes too. Yes, yes. I will answer the question at the end of this paper. Anyone, could you say, do you have lectures at 11:15 today?
Yes, sir.
Yes, sir.
11:15. Okay. Then I will call you later. We will finish this one. Okay. Practical also you have to study a little bit. But up to today, what you have learned from the practical botany class and this one, you will have the exam. But probably better to have not the 1st October, the 2nd October, Saturday, because it's better to have the exam the early morning because there's a net problem. If you conduct the exam, okay, this one, I will come. So DNA, I think DNA, you know this one will start from here. It's like this one, I think. It's already the small things you have already learned. What is DNA? It's everything, everywhere it's there. Okay, in the practical the quiz exam is better to have the early morning because if you want to have that this time, Friday, 10:30, it will be very difficult to have the Wi-Fi connection and this one. So better to have Saturday morning, 7 or 7:30. Before 8 o'clock, we have to better to finish. So I will put the exam date the 2nd October morning around 7 o'clock. Maybe the half an hour exam, just 25 quiz exam. Maybe very simple. At the meantime, if it is possible, you learn laboratory techniques also. I will confirm next Monday, or anyone you can ask me about this one. So, okay, there's DNA, deoxyribose acid. This is too small to see under the microscope. It looks like a twist of ladder. Every living thing has DNA, isn't it? Even from the virus, even the bacteria, fungi, and every living thing, it has DNA. That means that you have something in common with the zebra, tree, mushroom, and beetle. So I wonder the nature is, even you can see there's a bacteria and virus, there's a lot of difference. There's a clear difference, bacteria and virus, or bacteria and fungi. Even the microorganism and animals. There's each and every animal, cat, dog, cow, cat, goat, or everything is 100%. There's a clear difference there. Animals and tree, that's an exact difference. Human and animal, clear-cut difference. Animal and plants, big difference. There's no connection. But you remember, all these living things are totally different from each and everyone. But their DNA is exactly same. I wonder about this one. Even the bacteria, the DNA in the bacteria, DNA in the virus, DNA in the animals, DNA in the plants, DNA in the human being, DNA in the shark, DNA in the dinosaur. Exactly same. How is? I really wonder about this one. Each and everything is different, but DNA of all the living organisms is same. So that's what we are going to study. That's a important thing. Ever think about this one? Okay, we'll come. Then even though your saliva and here, you can get it this one and you can see under the microscope. Every living thing is made of cells. I mean, somebody, excuse me, sir. We will get what we learned in the practical sessions through for the quiz exam, etc. Yes. Practical exam also. Don't worry about that. Don't up to now until this week. No, not until next week. Practicals no need to study for the exam. Until today, what you have learned. If it is possible, please go through this one also. Laboratory technique also. You can have it. Don't much worry about exams. But if you continuously put it in the chat box, I can't do it anything else. Excuse me, sir. Will we get what we done? Thank you, sir. Okay. Until today, what you have learned from the cell biology and from botany, please go through. You have to practice. You see, okay, I will complete. The DNA stands for deoxyribonucleic acid. This chemical substance is present in the nucleus of all cells in the whole living organism. DNA control all the chemical changes which take place in the cells. The kind of cell which is formed, even the muscles or blood or nerve or in human, is controlled by the DNA. The kind of organism which produce whatever in a human is controlled by the DNA. All its functions and everything is determined by the DNA, even the shape, function, and everything. That is dependent on the DNA. Okay. I don't know. Especially, we are seriously worried about yourself. Even now, some of the students, they are told we are very stressed exams and each and everything. Even therefore, already we have in the faculty, we have the student counselors. They are 2 3 student counselors in that country. I think they have contacted you. Maybe Roshamis or somebody else. Or they take, if you want anything, want to do the academic thing, you can control. Again, another thing, they put the mentoring. If you have any problems, you can ask we will decide the mentors. Okay. Even I am the person for the mentoring also. Okay, later we will, I think thereafter we will have another session. Okay, DNA is very large molecule made up of long chain of subunits. Okay, what is within the DNA? What is we call? The subunits are called nucleotides. I think you have heard about these names in the A level. Anyway, we'll come. Each nucleotide is made up of a sugar called deoxyribose. That's the one sugar. So DNA is made up of a nucleotide. So within the nucleotide, there's a sugar. 3 components are there. One is sugar. What is sugar? Like a sucrose, maltose, fructose. It's the ribose. But there's a lacking of oxygen. One oxygen. Therefore, deoxyribose. So in the DNA, they are made up of the long chain. There's a very large molecule made up of long chain of subunits. Those subunits we call nucleotides. Within that nucleotide is developed by the 3 components. One is sugar. like a fructose, sucrose, like ribose. One. Then another one is phosphate group. One sugar, that is deoxyribose sugar, then phosphate group. Then one more thing that is may change the organic base. There's the nucleotide, there's four different organic bases are there. I think you have heard about adenine, thymine, cytosine, guanine. It may differ in each nucleotide, but there's the deoxyribose and phosphate group are same in all the nucleotides. Okay, okay. These are important things we have to learn. Okay, even the A level, how many? Just imagine the chemistry. How many books and how many notes? 2.5 years, more than 2.5 years, learn and sit for the one exam. But when you come to the university, I don't know, we are taking more care with you. You give them just 5 hours lectures, notes. Within that 5 hours, you will ask which section, sir, we have to learn. Not only, I don't blame you. Even the final years, last week, I gave one on the A4 sheet, printed one, 4 sheets. Just 4 sheets. I will, the quiz exam will come from this 4 sheets. Then they will study, I have explained each and every word. Then only I told the exam will come from this one. The previous day of the exam, they told, so we are very hard, so we can't memorize these things. It's very difficult. So please, I don't know. The A level simply you are studying. Just I think this is maybe the 5th lectures. My one. Just 5 hours lectures. You are going to have the exam, but you are wondering. I don't know. So okay, just try. Don't worry. Then the ribose is a sugar like a glucose, but with only 5 carbon atoms in its molecule. And deoxyribose is almost the same, but lacks oxygen. Both molecules may be represented by the symbol. I think this symbol you have studied in the organic chemistry, isn't it? Ribose and deoxyribose. The most common organic bases. Then phosphorus is same. The organic bases I have to adenine, thymine, cytosine, guanine. Later you will learn one by one. Just I will check the time. So these are the things. That's no need to shape. That shape is because I put in this one always when it's adenine because double helix, no double stand. So if it is this stand adenine, then thymine will always join. If it is cytosine, guanine will join. Therefore, I put it shape. No need to write that shape. Then the deoxyribose, the phosphate, and one of the base, adenine. This base may be this. Everything is same in the nucleotide, phosphate group and deoxyribose, but this base may be adenine, thymine, or cytosine, or guanine. They combine to form a nucleotide, so they will join together. This is the nucleotide. Then they will join in a chain. Isn't it? Nucleotides. Okay. If you have any problem, you can tell me about the exam or anything. Just we will have try. If this problem, then we will have. We'll think about just continuous assessment. The continuous assessment you have to have quiz with semester term paper presentation. I think term paper presentation. You can go and prepare this one. Okay. So that's nucleotide. The joint nucleotide. Thereafter, each nucleotide, see this nucleotide, they will join in this way. So will come. So the joint nucleotide, they will join and make a chain. The molecule of DNA formed by millions of nucleotides join together in a long chain. It will become a long chain. How to measure the size of this one, later we will study. So like this one. So please note it something, whatever is possible, you write it, get the notes, and even if you don't write this one, please note it that one simply. Phosphate or deoxyribose. And for the quiz exam, don't worry about much thing. We will have the 25 question. Maybe sometimes if you learn, you will finish within the 10 minutes. If you know the answer, even 10 minutes is too much. Within the 10 minutes, you will finish. Just half an hour exam. Don't worry about that one. You try to learn these things. Here, what are the things we learn? Prokaryotic, eukaryotic, animal cell, plant cell, bacteria, virus, then plant cell wall. Now this thing we are trying. That's all. Don't worry about serious. Don't make it you like don't worry about like A-level exam. Case everything is easy. But the thing is, A-level exam, the A-level exam, you have a competition. You have to compete and whether you have to get. But here, you should, maybe you have studied in the A-level, but here, don't study. Learn something. That's important. University, don't study. You don't teach. You shouldn't teach. You have to learn something. For example, what's the difference between studying and learning? If you want to swim, you can study, okay, how to swim, take the dress and put in the swimming dress and put the glasses here and put in the cap. Here you can learn the first step. Change the shirts, put the clothes and put the step through the goggles and set free the cover and everything. Then step 4, you get into the light this shape and move the hands and move the legs. That is study. Learning means you have to get it in the water or there. You have to try to swim. You get it. So, in fact, DNA usually gets the double standard nucleotide. The sugar phosphate chains are one of the strands are pulled together by chemical bonds between the bases. So you can see there is okay. They put it set off. So other cell organelle will come with section. No, other cell or other things until today, but we have learned from the Department of Agriculture Biology. Another from botany cell biology. That's it. That's all. Don't worry about that one. It will make it more simple. Don't worry about the exam. Okay, what have told? Okay, this is DNA. We will join last what I want to say. Okay, here they will like a chain. So if it is this one, so you can see how they are making the double stand. The single stand there. Oh, why not you go? So new students are coming today also. One person come. So I think it is better to conduct the quiz exam October 2nd or 2nd or 3rd week. But we not after previous stuff because 4th week come up. Okay, you can wait, it's okay. But the problem is, even I have joined when in the faculty after the 12 weeks. Already 10 quizzes finished, 10 department, 10 subject quiz finished, 10 subject midterms are finished. The term of presentations going on. Thereafter I came the 12th week. Then within a week they have told we have to start next week, we have to write on your quiz. 12th week I came, the 13th week I attended all the quiz exam within a week. And the 14th week I have attended all the quiz exam. And 15th week I have completed all the term of the presentation. It's my risk. So, they can in the 1 week now. They can go through the new notes. Okay, anyway, we'll tell them on Monday we will discuss about. So here you can do standard DNA, then they will become a double helix. So we will write something. Yes, you can go through, you can see My voice is from now on. Okay, okay.
We can hear you, sir.
Okay. Because of the internet problem. Is it okay now? Is it okay now?
No, sir.
Okay.
No, sir.
Yes. Just wait. Is it okay now? I don't know. Suddenly I didn't do anything. Can you hear? No. I, huh? Very. Hello?
Yes, sir.
The newcomers you can say, but I have told, I when the I was the first student and this was senior. I have made the now the professor, but really I was a senior at the fourth bench and first professor from this faculty. I'm the first professor, the one and only professor from this faculty so far. Uh, I joined after the 12 weeks and they told there's 3 more weeks left to finish for this exam quiz semester from the whole departments. Then I did the 13 week all the quizzes, 14 week all the semesters. So you have to do something. We can't wait there. Even some of us when they will come after. That's what simply I have told. I after 12 weeks. So we can't wait for the each and everyone. That's what I am telling now. Newcomers, when you come, just start. But anyway, I can wait for but I have simply told I came after 12 weeks of the exam. That first semester, when I come to join this as a newcomer in the faculty, was already that's a 12 weeks finished. 12 weeks finished. Simply I have told, they have told if you want to follow, they told, no, it's already quiz exams are finished, you can't follow.
Sir, we can't hear your voice.
Okay, that's the problem.
Voice is not clear, sir.
Okay, thank you. I have already started during that one. I did not do anything. Is it okay now? Could you hear me? Could you hear me?
Yes, sir, we can hear you, sir, but it's a noisy. It's okay, sir.
Really? I don't know what's the thing. Just we will move this one and you will get the response. Okay. Anyway, in case of exam, we will think about that one. Better to not wait even after 10 weeks. Some of the students will come. So for them, we can't wait. Just we will have no worrying on this one is too much of syllabus. At least we can have several techniques. Monday you will tell me. I will discuss with Roshan. So this one you can see added in our forms one with the timing. So they make double bond like this one. Newcomers, don't worry. If you are already late, then why you are want to wait for some other things? Immediately you start. The cell biology, the newcomers, we have studied the prokaryote, eukaryote, the basic plant cell and structure of the plant cell, animal cell. That's the difference. Then the bacteria and virus. Just five lectures, no? 5 hours lectures. Can you pick up with the delay and able but want to go very slow? It means everything you will do like that one. But if you ready to mingle with the present situation, that will be better. Just 5 hours lectures. Day level, how many hours you have studied the tuition? Chemistry. I am going to have a lecture, the 5 hours lectures, maybe the practical, maybe even 5, I think maybe 4. Including this one, maybe 5. Not more than 5. Even the practical also, just 5 practicals. Isn't it? You can't, even the newcomer, even the student can come on next Monday. Within the 5 days, are they can follow the 5 hours lectures, the 5 hours, 5 practicals? But you want to tell the reasons. You can tell. Always you can tell. So shall we have the exam through the online? No, sir. You can say 1,000 reasons. So network problem. So I have run a very small phone. So I am not a newcomer. So it's we don't have notes. You can tell. So 1,000 reasons. But don't try to find out the reason. You find to get it that one. Okay. Well, there's a pairing of DNA. I will explain the next class. They will come. Okay, now we have learned this is everything. This is a nucleotide, the phosphate and sugar, then organic base. Then they will join, that is one nucleotide. Then nucleotide, they will join and make a long chain. Thereafter, they will pairing up with another one and they will make double helix, double strand, DNA, double strand. So this paired strand are coiled into spiral called double helix. These are the double helix. These are basic things, but very, very, very important. So you can see this one. The double helix. You can write it. This one. The double helix. The sugar phosphate chain in between the bases. Later we will learn how to measure the length. From this one to this one, we got one base pair. BASC, base pair. That's like let's say when to measure the length centimeter or meter or liter or gram, like that. It's the base pair. 1,000 base pair, as usual, 1 kilo base. So from that one, we will stop here. I think we may have another lecture. So we will stop here from the basic. Next class we will see the replications, self-replication, how it will replicate. Before the cell divides, the DNA strand unwind and separate. Each strands make a new partner by adding the appropriate nucleotide. The result is that there are no true double-stranded DNA molecules in the nucleus, so that when the cell divides, each nucleus contains identical DNA. This process is called replication. So during the cell division, the double strand will become a single strand, and each single strand they will form their pair and become a true double strand. That is cell replication, DNA replication. We will study in the next class. So next Friday we will have the lecture. we want to have the exam, but next Friday lectures no need to go the exam. Next week practical also not necessary. I think if you want to go in other lectures, you can leave. We will study the DNA replication in the next class. Okay, but it's difficult to understand anyway. We'll think about the exam on Monday. I will tell you, don't worry. Okay, you can leave.
Thank you. Thank you.